We're going to continue our discussion about enzymes. So in the previous video I mentioned that I will be talking about activation energy in a little bit more detail through a diagram, and this is what I have up here. So on the x-axis I have the reaction progress of a specific reaction, and on the y-axis I have the delta G. So the black line is the reaction in the absence of an enzyme or a catalyst, whereas the pink line is the reaction in the presence of a catalyst or an enzyme. So you can see that the difference between the black line and the pink line near the hill region, where it says A, it's quite decreased. So what happens is that an enzyme, it'll lower that activation energy so that not a lot of energy is required for the reactants to be converted into the products. The T at the top represents something called a transition state. That is where half the bonds are broken and half the bonds are formed, and the reaction can basically go either way. If there's enough energy, it'll go to the products. If there's not enough energy, it'll just come back to the reactants. So the lower the hill, or the lower the activation energy, the faster the reaction will go. So this is what enzymes basically do. They just lower the activation energy by either facilitating a more conducive environment, facilitating more collisions, or anything of one of those mechanisms that help the reaction go to products, or even the reverse if it's more favorable in the reverse direction. So some of the mechanism of actions of enzymes are either an induced fit or a lock and key. These are basically the two models that are out there, but I would say that the induced fit model is more acceptable, just because it just makes a little bit more, there's more evidence towards it. So what the induced fit model basically says that is that the enzyme and the substrate, and substrate is just a fancy word for the molecule that's going to be attached to the enzyme, or it's going to bond with the enzyme. So the enzyme and the substrate kind of have like complementary shapes. They're kind of, similar to each other, but they're not necessarily perfect. But once the substrate binds to the enzyme, both of them kind of undergo kind of like modifications and better bond with each other. So that's what the induced fit model says. What the lock and key model says is that it's kind of like a lock and key. There's only one key that fits one lock. So it's kind of like this rigid structure. It's either an all or nothing kind of phenomenon. So it's kind of like a force or a perfect fit that this model is suggesting. Once again, the induced fit model is more accepted in the scientific community. Epoenzymes and holoenzymes. So what these are is, if you remember from the previous video, I talked about prosthetic groups, which are basically just covalent attachments to proteins. They work better in the presence of prosthetic groups, which can be vitamins or metal ions or lipids or carbohydrates or anything of that sort. So if the same thing goes for an enzyme. If an enzyme has a prosthetic group, it's called a holoenzyme. And if it doesn't have a prosthetic group, it's called an apoenzyme. The way I remember it is absent and apo both start with an A. So the prosthetic is absent, and that's what I wrote down over here. And to give you a better idea of the induced fit and lock and key model, was what I did is kind of drew like this really rough diagram. So one represents an induced fit. So you kind of see like over here, it's kind of in the same shape, but it's not exactly the same shape. So once these two bind, they kind of facilitate each other better. They kind of change their conformation a little bit so that they can bond better. Whereas over here, you see that it's an exact fit. Like it just fits perfectly, and there's no other. It's really rigid. It's not really flexible, and it kind of just summarizes the lock and key model.